I'm Dr. Anjani Devi, Associate Professor, Department of Biotechnology, Vignan's Foundation for Science, Technology and Research. I'm handling the course Enzyme Technology. Before we start the course, we need to know what are the topics involved in the first unit of our enzyme technology. Here, in this enzyme technology, in the first unit, we are going to learn about discoveries of enzymes, classification and application of enzymes, principles of catalysis, role of entropy in catalysis, comparison of chemical and enzyme catalysis, stability, deactivation and catalytic activities, mechanism of enzyme action, concept of active site, and specificity of enzyme reactions. And what are the outcomes of this course? If we are going to learn this course, what we'll be learning at the end of the day? We'll be gaining the ability to describe structure, function, and the mechanism of enzyme actions. Moreover, we are going to model kinetics of enzyme catalyzer reactions and enzyme inhibitory and regulatory process. We can summarize current processes involved in industrial enzyme production from plants, animals and microorganisms, and we can adopt different immobilization methods and analyze the bioconversions in immobilized reactors. So now we are going to move into the actual topic. Before we start our enzyme technology course, first we need to know the definitions of certain important words. What is the one of the most important word which you have to know about is what is an enzyme and definition of an enzyme. What are enzymes? Enzymes are biological catalysts. That means they will guide the reactions that are taking place in a living organism. And what is the nature of enzymes? These enzymes are mostly proteinaceous in nature, but some enzymes are having RNA as their material, genetic material. That's nothing but ribozymes. And what these enzymes do? These enzymes will increase the rate of chemical reaction without bringing any alteration in chemical equilibrium of the reaction. After knowing about enzymes, we need to know about what is enzymology. Enzymology is the study of enzymes, their kinetics, structure and functions as well as their relation with one another. And now we are moving towards enzyme technology. What is enzyme technology? What this enzyme technology deals with? Enzyme technology deals with alteration in the enzyme structure, modification in the catalytic activity of enzymes in order to get new metabolites. And moreover, this enzyme technology is a multidisciplinary area which is comprising of microbiology, biochemistry, molecular biology, molecular genetics and biophysics. Why we need enzymes? If you consider any human body, it is made up of number of living cells and each cell is a site of number of tremendous biochemical reactions which we are calling it as metabolism. So this metabolism, this is a process of chemical and physical changes which goes on continuously in a living organism. During this metabolism, number of new cells are formed, number of old cells are replaced with new cells, and the food we are eating is getting converted into energy. We are disposing or releasing the waste material from our body. The reproduction, etc. All these are part and parcel of metabolism. And what will happen without enzymes? If you consider the reaction without enzyme, this reaction will be taking place very slowly and it will be taking more amount of time. But in presence of enzymes, these reactions are taking place very fastly. That means these enzymes are responsible for increasing the rate of reaction. And now, I'll give you an example. That's nothing but just consider oxidation of fatty acids into carbon dioxide. Just consider the oxidation of fatty acids to carbon dioxide and water. Consider this reaction under laboratory condition as well as in the human body. Under laboratory conditions, in order to complete this oxidation process, it will be taking enormous amount of time. Moreover, it requires high pH, high temperature and number of corrosive chemicals. But in the human body, this reaction is taking place very fastly at narrow range of pH and temperature. I'll explain you with another example. Just consider that we are going for complete breakdown of protein molecules. Under laboratory conditions, in 20% HCl, it is taking nearly 24 hours for complete degradation of a protein molecule. While in a human body, this process is getting completed under narrow pH and temperature conditions within 4 hours of time, and these reactions are taking place very rapidly and very smoothly. I'll explain you the action of enzymes by considering digestive system. See, a number of enzymes are involved in number of reactions that are taking place inside our body. Let us consider the digestive system. Just think that you are taking the food material. You're chewing your food material. When you're chewing your food material, your food is getting mixed up with saliva. And you know pretty well that the human body is secreting nearly 1 to 1.5 liters of saliva per day. And the saliva is having an important enzyme called as salivary amylase. This amylase will be acting upon the carbohydrate content of the food material, and it is breaking down that carbohydrate content into simple sugar molecules in order to get energy. And now, after the chewing process is completed, this food is entering into your esophagus. Through that esophagus, the food is entering into your stomach. Now, within the stomach, an important enzyme called as pepsin is getting released. Along with pepsin, HCl is also getting released. Now, your food material is going to mix up with pepsin. Pepsin will be acting upon the protein matter of the food material, and it will be breaking the proteins into peptides. Now, from stomach, the food is entering into your small intestine. In the small intestine, the enzymes from pancreas such as trypsin, chymotrypsin and lipases, etc. They are synthesized in pancreas and they are now getting released into small intestine. These enzymes are now getting mixed up with your food material. Trypsin is acting upon the proteinaceous matter of your food material, whereas lipases are acting on the lipid content of your food materials in order to break down that lipids into free fatty acids and glycerol. I want you people to see this video for proper understanding of the mechanism what I have explained you till now.
Chewing breaks down food particles and mixes them with saliva. Saliva contains the starch-digesting enzyme amylase and a slippery protein called mucin that helps to lubricate the food particles for easier swallowing. As food is swallowed, it enters the esophagus and is moved downward by wave-like muscular contractions called peristalsis. At the end of the esophagus, the food empties into the upper portion of the stomach. The lining of the stomach produces gastric juices that contain protein-digesting enzymes. Peristaltic movements of the stomach will mix and churn the food with the gastric juices. This action further breaks up the food particles, forming a milky fluid known as chyme. At the bottom of the stomach, a muscular valve controls the release of chyme into the first part of the small intestine called the duodenum. The duodenum is only 25 centimetres long, but most of the digestion takes place in this region. Also opening into the duodenum are ducts from the pancreas and the gallbladder. These allow enzyme-rich pancreatic juice and bile to be released and mixed with the chyme. Chemical breakdown of carbohydrate, protein and fats into smaller absorbable molecules takes place. The next section of the small intestine is the jejunum, about 2.5 metres long. Folds in the wall of the jejunum greatly increase its surface area, allowing for ready absorption of the products of digestion. The inside of the jejunum and ileum are lined with tiny finger-like projections called villi. These increase the surface area of the small intestine. They play an important role in absorbing the breakdown products of digestion. Each villus has a rich blood supply, allowing nutrients to be readily absorbed and transported to the liver. Also present is a system of vessels known as a lacteal, which absorbs fat nutrients and carries them to the circulatory system. At the end of the ileum, a muscular structure controls the release of undigested material into the first part of the large intestine, the cecum. From here, peristaltic movements push the undigested material down the large intestine and allow it to mix with the large bacterial population present. The bacteria are able to ferment some of the undigested material, producing short-chain fatty acids. These are used as an energy source for the bacteria and the cells lining the large intestine. Other important chemical compounds, such as vitamin K, are also produced by this bacterial action. As the material slowly moves down, undigested material is compacted and absorption of water and electrolytes takes place. Undigested and compacted material enters the rectum for temporary storage before being eliminated through the anus.
Now, we can conclude that our life is impossible without the presence of enzymes. With these basics, now we are moving towards our first topic, that is nothing but discovery of enzymes. Number of scientists have contributed in the field of enzymology. We'll just recall the history. Juban Fat is a scientist who have first extracted the malt from barley. In 1833, Payen and Perzen, they have extracted enzyme diasterase from malt extract. Now, the work of De Saussure. De Saussure have prepared a similar sort of compound as diasterase from wheat. Schwann is the person who have worked upon trypsin and pepsin. He have extracted those two enzymes from digestive system. In 1837, Berzelius have first given the concept of catalytic nature of your protein. That's nothing but enzyme. Pasteur, in 1857, he have proposed alcoholic fermentation and he have given the importance of yeast in alcoholic fermentation process. Now we are going towards what Kuhne have done. Kuhne is a scientist who have given the word enzyme for the first time. In Greek, enzymos means in yeast. In 1897, Buchner have given the name zymase to the enzyme which is catalyzing or which is fermenting sucrose. For his action, for his work and his contribution towards biochemical research, Buchner have been awarded with Nobel Prize in 1907. In 1926, Sumner have established the true nature of enzyme as protein. He have extracted urease from jack bean and he have crystallized it. See the crystals here. And for his work, Sumner was awarded Nobel Prize in the year 1946 along with two eminent scientists. Those are Northrop and Stanley. These two scientists have worked upon pepsin and trypsin along with chymotrypsin. Apart from these eminent discoveries, we have to learn about few more discoveries made by eminent people. In 1890, Fischer have given the lock and key model of enzymatic action, whereas in 1913, Michaelis and Menton have given the mathematical model for enzyme action. Pauling in the year 1948 have proposed transition state theory of enzymatic action. Sanger in 1953, he have determined the amino acid sequence of protein insulin. Lerner and Schultz, they have developed catalytic antibodies. I want you people to go through these links in order to gain more knowledge about this topic. Please do these questions, work on these questions in order to know how far you have understood today's lecture. Thank you.